Hello, I am Eve.
Hi, I'm Eve too,
We are human clones.
and we are going to teach you about cloning.
The term cloning describes a number of different processes that can be used to produce genetically identical copies of a biological entity. The copied material, which has the same genetic makeup as the original, is referred to as a clone. Clones can happen both naturally and can be made artificially.
In nature, some plants and single-celled organisms such as bacteria produce genetically identical offspring through a process called asexual reproduction. In asexual reproduction, a new individual is generated from a copy of single cell from the parent organism. Natural clones, also known as identical twins, occur in human and other mammals. These twins are produced when a fertilized egg splits, creating two or more embryos that carry almost identical DNA. Identical twins have nearly the same genetic makeup as each other.
There are three different types of artificial cloning: gene cloning, reproductive cloning, and therapeutic cloning. Gene cloning produces copies of genes or segments of DNA. Reproductive cloning produces copies of whole animals. Therapeutic cloning produces embryonic stem cells for experiments aimed at creating tissues or replacing injured or diseased tissues.
Question is, can we clone full humans by using any of these cloning methods?
Dolly is the first mammal cell cloned successfully from an adult cell. She was born in 1996 after scientists figured out how to remove the DNA from the egg cell of Scottish blackface sheep and basically replace it with the DNA of memory cell from a twin dorsal sheep. They gave it a little electric shock to fuse the cell and get it replicating, placed the cells in the uterus of another sheep and got the clone. Dolly was created by a reproductive cloning method, which could be theoretically used on humans, but this is the best case scenario. It took 277 tries for scientists to get one Dolly. Nowadays, cloning mammals generally has a success rate about 10 to 20%, better than 1 in 277, but still a majorly inefficient process. Scientists have been cloning other animals like frogs, mice, rabbits for years. After Dolly, two monkeys have been cloned using the reproductive cloning method that produced Dolly the sheep. This was the closest mammal to humans. Identical long-tailed macaques Zhongzhong and Huahua were born in 2018 at a laboratory in China. Scientists say that populations of monkeys that are genetically identical will be useful for research into human diseases. For a time, it seemed possible that human cloning had been accomplished on December 27, 2002. Bridget Boisler, chief executive of Clonate Company, held a press conference in Florida announcing the birth of the first human clone called Eve. It's been 18 years and we haven't gotten any proof that baby Eve exists or has ever existed, but the company is still alive and well. If science has done so good with cloning plants and animals, how close we are to being able to create a human clone? Comes out that there are more than a few barriers on the way to full human cloning, such as ethics, resource constraints, and trial and error costs.
To this day, there is an unsolved debate around the ethical factor of human cloning. A lot of organizations oppose reproductive cloning, considering it highly unethical. There are serious ethical concerns about possibility of harvesting organs from human clones in the future. Article 11 of UNESCO's Universal Declaration on the Human Genome and Human Rights asserts that the reproductive cloning of human beings is contrary to human dignity, that the potential life represented by the embryo is destroyed when embryonic cells are used, and there is a significant likelihood that cloned individuals would be biologically damaged due to the inherent unreliability of cloning technology. Ethicists also think that human cloning might change the shape of family structure by complicating the role of parenting within a family of convoluted kinship relations. For example, a female DNA donor would be the clone's genetic twin rather than the mother, complicating the genetic and social relationships between mother and a child. In other example, there may be expectations that the cloned individuals would act identically to the human from which they were cloned, which could infringe on the right to self-determination. Although it would be unethical, experts say it is likely biologically possible to clone a human being. But even putting ethics aside, the sheer amount of resources needed to do it is a significant barrier. During experiments, researchers encounter problems that need to be overcome with trial and error. With mice, researchers were able to use thousands of eggs and conduct many experiments to work out these problems. It's all about big numbers that the scarcity of human eggs does not allow. The practice of animal cloning cannot be directly mapped to humans. For instance, cloning an animal requires that researchers first remove the nucleus of an egg cell and with it the proteins that are essential to help cells divide. In mice, this isn't a problem because the embryo that is ultimately created is able to make these proteins again, but humans aren't able to do this.
What's more, cloned animals often have different kinds of genetic abnormalities that can prevent embryo implantation in the uterus or cause the fetus to spontaneously abort or the animal to die shortly after birth. These abnormalities are common because cloned embryos have just one parent rather than two, which means that a molecular process known as imprinting does not occur properly in cloned embryos. Imprinting takes place during embryo development and selectively silences certain genes from one parent or the other. Problems with imprinting can result in extremely large placentas, which ultimately leads to problems with blood flow for the fetus. In one experiment, scientists cloned a species of cattle called banteng, and it was born at twice the size of a normal banteng. It had to be euthanized.
Given all this, we can say that cloning humans, although scientifically possible, has many more factors to be considered rather than only durability. So for now, maybe the idea of cloning entire human body and organisms is not as close as we think, until science unravels ways to bypass the many risk factors and the ethics issues of the human cloning. Until then, I am just Eve without Eve too.